My name is Eric Walters, and I work in the expression and purification of gene M with my mentor, Dr. Jason Hurlbert. Now, gene M is a virulence factor of the plant pathogen Clavibacter michiganensis, which is gram positive, meaning that it has a thick peptidoglycan cell wall, allowing it to exist both inside and outside of plants. If you look to the right, you can see the ring rot that it causes and necrosis that it has caused in a tomato plant. Now, the thing about this bacteria is really easily spread. And so if you have a bacteria that's easily spread, infecting a lot of crops, you have a large crop loss, and that has a profound financial impact on both your producers and your consumers. So it's pretty important to figure out what this virulence factor is doing. So gene M, or the mystery gene, was identified by our collaborator at the University of Florida, Dr. Frank White. It has been shown to cause symptomatic necrosis in both tomato and potato plants. Now, the interesting thing to look at if we're examining the amino acid sequence is that if we mutate this valine to an alanine, gene M is no longer symptomatic for necrosis. And it's important to note that this is a symptom of the function, not the function itself, because the function is unknown. And this protein is right around 17 kilodaltons. And so our job is going to be to identify the structure and function of the expressed protein. Now, we do have some hints from our collaborator, Dr. Frank White. He believes that it might be a patatin, and upon further inspection, it could, because we do have the serines and aspartic acid residues, as S and E denote, that would participate in the catalytic dyad for the phospholipase activity of patatins. But it also doesn't have cysteines, which is very common in patatins for the conserved cysteine mechanism that they are involved in. So we split this research up into two phases. One was the bioinformatic analysis, and the second was just lab research. And the thing I really want to focus on the bioinformatic analysis in the interest of time, since we can't really cover all of this, is our modeling with TR Rosetta. TR Rosetta is an interesting program that doesn't use the protein data bank like other homology modeling software programs, but rather it creates it from new. What it does is it kind of looks at each of these individual amino acid residues. It may look at possible H bonding distances. It also looks at the location of beta carbons relative to the amino acids. And it's able to kind of go along and look at different lengths of amino acids to predict a structure for your protein. And if we look at this model, we see what we should see with a real protein or a good protein model. We see tight folding. It's very tight in here. And we see alpha helices, beta sheets, this defined secondary structure. And we have a 79% confidence. And what we know is that above 70% confidence means that we have a homologous molecule to our target protein. And so if we look over here, we see this unordered area, which corresponds to this part of the protein. And this is a terminus that is pretty common with DNA binding proteins, where they have this intrinsically unordered area, which allows it to bind to multiple different partners. So maybe it's an intrinsically disordered protein and it binds to DNA. And this is one of the things that bioinformatic analysis help us kind of think about. And this model is also going to be useful later to solve the phase problem that is very common in when we do X-ray crystallography to find the structure of the protein. Now, to start our laboratory research, the best thing we can do is make sure that we can express gene M using recombinant E. coli. And all of these should be protease deficient, so it doesn't chew up your protein. And so we use BL21, Rosetta 2, and Nico 21. And essentially what we do is we have a plasmid that has gene M and it's cloned into our E. coli. And on that plasmid, what we have also is a repressor, and that repressor will not express gene M until IPTG, or the inducer, is added. So what we can do here is we can take gene M, grow it up in culture, and then create overnight cultures and eventually inoculate, right? And we can induce one and not induce the other. And what we should see here is that the induced variant should show protein, because once again, IPTG is added, which is going to remove that repressor, and we should have expression of gene M. And we're looking for the one that's going to most strongly express our gene. And so how we analyze this is SDS page. which gives us weight, purity, and amount, but it doesn't identify it, but it can give us a protein in the right weight. And so what we saw as a result of this is that as we go along this 17 kilodalton area, we don't really see much expression in these others two, but right here, we see very strong expression in the induced versus the uninduced form at the 17 kilodalton region. So what that tells us is that we should use E. coli Rosetta 2 for further expression and purification trials. So what we're going to do is we're going to go ahead and scale up with Rosetta 2. So we grow up a large 6 liters of culture. We lyse those cells, extract our proteins, and we can develop a purification method that's going to use chromatography. And our first chromatography step is metal chelating affinity chromatography. The interesting thing about this protein is that we have engineered it so that it has a 6x histidine tag on it, which means that it has 6 histidines in a row, which is not biologically present in any other proteins. And so what that does is that it allows us to bind to this column, which has a 2+ metal, because histidine has an N with 2 electrons. So it can bind to the metal column, and other proteins that can't bind will just get washed off. Next, after that, if we want to remove our protein and elute our protein out, we're going to apply imidazole, which is exactly the same as histidine, but it has more degrees of freedom because it's free imidazole. And so it's able to push off our protein because it's more entropically favorable. That binding is more entropically favorable. And what we see here is a protein peak as we apply imidazole. And so we investigated this with SDS page. And what we see is we have a protein at 17 kilodaltons, but we don't know if it's our protein, but we do know that it's got a lot of other junk or heavier proteins in there. So what we think we can do is go ahead and do gel filtration and filter that out. And so that's the next thing we did was gel filtration, which basically big proteins kind of wash out. Small proteins get stuck in these little beads, gel beads, and they have to navigate through and slowly elute. And that's why it is able to separate by size, because the smaller it is, the later it comes out. And we're going to go ahead and investigate those peaks. And so here's the gel SDS page for our gel filtration. And what we see here is, once again, we have a protein at 17 kilodaltons, but it is remarkably more pure than the other, and we don't have a lot of other contamination in there. So the last step would be to try a cation exchange, which is maybe they have different isoelectric points, so maybe we can separate them out. So we use cation exchange chromatography, which essentially applies a positive charge to our protein by selecting a buffer that is at a pK or a pH below our pK of our protein. And that gives it a positive charge. And so that positively charged protein is going to bind to a negatively charged column. And to elute it, we're going to apply salt and it'll elute out. And so once again, we do see a peak, but we have to investigate that. So rather than showing you the SDS page, I'm going to show you the Western blot. The Western blot uses antibodies to detect a histidine tag specifically. So it has a primary antibody that detects our histidine tag and a secondary antibody that binds to the primary. And then we have a source that is going to allow it to give us a color. And so here we have, once again, gene M. We can say this is definitely gene M because it is verified by Western blot, which the antibodies would not bind, and it's very pure. So we have our pure protein. So what we can say is now we have gene M has been expressed by Rosetta 2. We saw that by the first SDS page. Gene M has been purified in 3 steps, which we have MCAC, cation exchange, and gel filtration. And then finally, our protein is ready to be characterized because we had 2 mg per mil of protein purified. But we need more protein, about 10 mg per mil, to crystallize. So our future work is a phospholipase assay. So since we think it's a betatin, what we're going to go ahead and do is we're going to use a phospholipid. We're going to cleave it, which is going to give us linoleic acid. And then we're going to, once again, oxidize this to create a linoleic hydroperoxide. And that should absorb at 234 nanometers. And that'll tell us if it's a betatin. So I would like to acknowledge my mentor, my lab members, Maiden SC, the University of Florida, the McNair Scholars Program, and the NSF.